Good morning, everyone. Good morning. Thank you, Amelia, Brianna, Chelsea. Thank you for signing in. If you would, go ahead and type your first name or your nickname into the chat for me. Thank you very much. Good job. Good to see everybody. So today we are going to be moving forward with our experiment in oxygen requirements and growth. And then we're going to begin our next experiment for the week, which is about the use of ultraviolet radiation to control the growth of microbes. Before we get started in that, does anybody have any questions about what we talked about last time, or anything that you might be learning about in lecture? If you have any questions, feel free to type them into the chat, or you can also unmute yourself and speak out if you'd like.
I emailed you about a question. I don't know if you read it. It was just a few minutes ago. I think it's question 4 on the quiz. I think it might be wrong, like the answer to it.
Is that the microbial growth quiz?
Correct. Yep.
I didn't get your email, Brooke, but I will check on that after class for sure. Okay. Thanks for bringing it to my attention. Periodically,
no
when I'm making quizzes, part of what you do when you build a quiz in Blackboard is you have to come up with, all of your possible answers, and then you have to mark the answer that's correct. And if I forget to do that, what Blackboard does is it just chooses the first answer as correct in my list. So I will double check on that and see what's going on with that one. Thanks for calling it to my attention. Very good. All right. Anything else? Okay. I do want to remind you that here, I'll let me share my screen with you for just a minute. I want to remind you that there is a content folder available for you on Blackboard about your pathogen assignment. If you click in here, you're going to find the instructions for this assignment, as well as the place where you're going to submit your first draft and your final draft. When you submit your first draft to me, I'm going to be going through it and making comments for you about where you can improve. And the assignment will be graded in 2 parts, the first draft and the final draft. This is due, your first draft is due on March 24th. That's 2 weeks from today, just a heads up. And remember, this assignment is about you choosing a set of questions, essentially, about different pathogens. There are several different pathogens that you will have the ability to choose from. So you can see question group 1 is about the pathogen that causes tuberculosis. And the second one is about cholera, and the third about Zika. So you get to choose which set of questions you want to answer for this assignment. So just something to, a heads up for you, something to remember that's on your calendar. And then looking ahead to next week, let me see if I can do this and not lose the screen here. Looking forward to next week. Just a reminder, this is a very unusual week for us next week, because you're going to have a lecture exam. Well, on Monday, we'll be meeting as usual for lab. And then on Wednesday, you'll be having a lecture exam. And on Friday, you'll be having a lab practical exam. So very unusual week for us, just to give you a heads up. All right. So the last time we were together, we started talking about oxygen and the requirements that certain microbes have for different oxygen environments. And we talked about the different categories of oxygen requirements for microbes. And we talked about how incredibly diverse microbes are in terms of their need for oxygen or their need for an environment that's free from oxygen. If you remember, we defined 5 different categories of oxygen requirement. We talked about obligate aerobes and obligate anaerobes, sort of 2 ends of the spectrum there. We talked about organisms that are called aerotolerant and micro aerophiles. And finally, the largest category, which is the facultative anaerobes. And we not only defined what their needs are, what their requirements are regarding oxygen, but we also talked about the types of metabolic pathways that these organisms tend to use in order to make their ATP. Because remember, when an organism requires a molecule like oxygen, it's because it's using that oxygen in one of the steps that are involved in making ATP. And oxygen happens to be what we call the final electron acceptor molecule at the bottom of the electron transport chain in the process of making ATP. So we talked about aerobic cellular respiration as one of these pathways to ATP, and anaerobic cellular respiration, and also fermentation. And we talked about how different organisms have the ability to use one or more of those pathways. And remember, the facultative anaerobes, of which most bacteria are able to use all 3 pathways. They're able to switch. So when oxygen is around, great, they'll do aerobic cellular respiration. But when oxygen's not around, well, if there's another final electron acceptor molecule that they're able to use, they can do anaerobic cellular respiration. And if there are no final electron acceptors available, well, they'll switch over to fermentation. So very, very adaptive these bacteria are. So before we left last time, we talked about this important media called thioglycolate medium or thioglycolate broth, and how we can inoculate this broth and then incubate it and then look at the growth patterns of the cells inside the broth. And it's not going to look like regular broth when it comes out of the incubator. Remember, we said for regular liquid broth, when cells grow in it, they grow dispersed throughout the broth, and the broth just gets cloudy top to bottom. But in thioglycolate, because we add a little agar to the broth, you end up with cells sort of growing in very discrete areas in the tube. And the cells kind of clump together at times, and they make sort of these thick mats of cells, and sometimes these long strands of cells. So we'll get to see those today as we look at our results. So I'm going to pull up our lab slides here, and that's what you should be seeing right now. Oxygen and growth. Remember, these were the 5 categories that we talked about. We talked about what the growth patterns look like in thioglycolate tubes. And we finished up, and of course, we also talked about this Catalase test and how we can use this very simple, very inexpensive test to determine if microbes have this enzyme called catalase. So we finished up right around here. We looked at 4 different bacteria that we will be using in our exercise today, that we're going to use to inoculate thioglycolate broth. So the first organism, remember, is Clostridium Sporogenes, gram-positive rod. Enterococcus Fecalis, which we've talked about before. This is a gram-positive coccus or coccobacillus even. E. coli, our old friend E. coli, gram-negative rod. And finally, a new one, Alkalogenese Fecalis, which is also a gram-negative rod. Two of these organisms live entirely in the body, primarily in the intestinal tract. And two of them can be found in the intestinal tract and outside in the soil and in marine sediments. So we inoculate broth tubes for each one of these organisms. I also said that we're going to streak these organisms out on just plain old nutrient agar. Remember, that's a basal medium. And we're going to streak 2 plates for each one, each organism. One plate is going to go in the brewer's jar and into the incubator. So that one will be in an oxygen-free environment. And one plate is going to incubate in room air in the incubator. And finally, we'll look at some catalase testing results for each of these organisms as well. So, as usual, when we do an experiment, we're going to take a couple of minutes to make some predictions. Okay? It's really important when we design experiments in the laboratory that we think about the things that we already know, and we distinguish those from the things that we're trying to figure out. And when it comes to oxygen categories and oxygen requirement categories, we can already talk about the things we know about what we would expect for each category. So for example, we can ask ourselves the question, what can grow on a nutrient agar plate? Remember, that's what Na stands for. That we incubate in the presence of oxygen. In other words, in the presence of air. What kind of organisms among our 5 categories are able to grow when we streak them out on this agar plate and tuck them into the incubator? Well, certainly we know that the obligate anaerobes are in that group. Remember, these are the organisms that require oxygen. They must have it or they will die because they use oxygen to make their ATP. Facultative anaerobes are in that group too because these organisms, while they don't have to have oxygen, they do not find oxygen toxic at all. It's not going to hurt them to be in an oxygen environment. And in fact, when oxygen's around, they can use it to make ATP. We also have 2 other categories that will fit here, but there are some exceptions to them. There are some things to note about them. And that's because both of these groups, aerotolerant and micro-aerophile, they have very specific requirements about oxygen, don't they? They have very specific requirements about how much oxygen they're going to be able to use or even just tolerate. There's one group that's not on the list, right? The obligate anaerobes are not on this list. If we put a nutrient agar plate into the incubator just in air, in room air, exposed to oxygen, the only group of organisms that will not be able to grow are the obligate anaerobes because they find oxygen toxic. Let's review aerotolerant and micro-aerophile for just a moment so we can remind ourselves about their very specific requirements regarding oxygen in the environment. Aerotolerant, as the name suggests, are going to tolerate air. They're going to tolerate it. In other words, oxygen is not toxic to them. It's not going to kill them, but they're also not going to use it to make ATP. And we said that they grow best in low oxygen concentrations. In other words, less than what is found in room air. So in an incubator that's exposed to just plain old room air, aerotolerance will grow, but they're going to grow poorly. They're not in their most favorite environment. If you look at the micro aerophiles, we see something a little bit different. Micro aerophiles, remember this word phile or philic means love. They love air, but in a micro amount. So they require oxygen, but they need it at a reduced concentration. This is the group that grows best in low oxygen and higher than normal carbon dioxide. So no surprise, both the aerotolerance and the micro aerophiles will grow in room air. They're just going to grow poorly. Now, what we mean when we say grow poorly, when you're talking about an agar plate, is that you'll see colonies growing, but they're going to be very small. They're not going to be anything close to the size of colony that we would expect for the organism. Now, let's look at the opposite situation. What will we expect for growth on nutrient agar plates that are placed in a brewer's jar where there's no oxygen? Obviously, the obligate anaerobe is going to grow in here because obligate anaerobes find oxygen toxic. So they're going to grow just fine in an environment with no oxygen in it. But remember, they're not the only ones that can grow in a brewer's jar. Facultative anaerobes will. Remember, they're the ones that are the most adaptive. No oxygen? Okay, they'll just do another metabolic pathway. If there's another final electron acceptor available, they'll use that. If there's no final electron acceptors, well, they'll just leave cellular respiration completely, and they'll do fermentation instead. Finally, aerotolerant organisms will grow in a brewer's jar. They're just not going to grow very well. They're going to grow poorly. Remember, they don't use oxygen to make ATP, but they grow best in a low oxygen environment. Now, let's think about catalase tests. Who should have a positive catalase test? Well, the organisms who use oxygen most often, who are making their ATP with oxygen. So that's the obligate aerobes, the facultative, oh, this should say anaerobes, sorry about that. The facultative anaerobes, remember, they don't always use oxygen, but if oxygen's around, they'll use it. And finally, the micro aerophiles, they're using oxygen to make ATP. They just don't like it at very high concentrations. These are organisms that routinely use oxygen, so we expect them to have oxygen byproducts floating around in their cytoplasm, and those oxygen byproducts have to be degraded, particularly hydrogen peroxide. And catalase is what destroys hydrogen peroxide. So who would we expect to have a negative catalase test? The other two categories, the obligate anaerobes and the aerotolerants. Now, obligate anaerobes make sense because they find oxygen toxic. So you're not going to find oxygen in these cells. So you're not going to find oxygen byproducts in those cells, and you don't need any catalase to break those byproducts down. But the aerotolerant might surprise you. Remember, aerotolerants grow best in a low concentration of oxygen. So it would be really normal for them to have oxygen in their cells. So how come they don't have catalase to deal with oxygen byproducts? And the answer is just these organisms have evolved so that they simply don't have the pathways, the metabolic pathways to generate oxygen byproducts. They just don't make oxygen byproducts like most other cells do. So they have no need of an enzyme to break down those byproducts. All right, so just a reminder about what these tubes are going to look like, at least in theory. Obligate anaerobes grow throughout the oxic zone. Obligate anaerobes grow at the very bottom, at the deepest part of the anoxic zone. Facultative anaerobes grow best in the oxic zone, but they can also grow throughout the rest of the tube. Aerotolerants grow in the lower portion of the oxic zone, as well as the rest of the tube. And micro aerophiles grow in the lower portion of the oxic zone. So we have these different growth patterns, and we should be able to pull these tubes out and see one of these or these patterns demonstrated for us, and it should give us information about what group each one of our organisms belongs to: the Clostridium Sporogenes, the Enterococcus Fecalis, the Alkalogenese, and the E. coli. We should be able to see which group they belong to based on how they grow in these thioglycolate tubes. So let's take a look at our results. First of all, let's talk about the nutrient agar plates. Remember, I spread cells for each one of our 4 organisms on 2 different nutrient agar plates, 2 plates for each organism. I took one of the plates and I put it into the incubator in room air. And I took the other plate and I put it into the brewer's jar and I put it into the incubator. So for the plates that were just left exposed to oxygen, exposed to room air, the Clostridium Sporogenes did not grow. Okay, the Clostridium Sporogenes did not grow, not even a little. The Enterococcus Fecalis grew, but it grew poorly, very small colonies. The E. coli grew, good-sized, normal colonies. And the Alkalogenese Fecalis grew, good-sized, normal colonies. Now, when we compare these results to the plates that incubated in the brewer's jar, they look very different. The Clostridium Sporogenes grew into good-sized, normal colonies. When it was exposed to room air, no growth. When it was placed into the brewer's jar, nice, normal growth. What do you think about Clostridium? If you had to guess right now which one of the 5 groups Clostridium Sporogenes belongs in, what would you say? Type it into chat for me. When we streaked a plate with those cells and put that plate into the incubator and left it there for 24 hours, nothing grew on the plate. But when we took that plate, well, a second plate, and we put it into a brewer's jar and took all the oxygen away, and we put the plate, the brewer's jar into the incubator and grew it for 24 hours, woo, plenty of growth. Normal sized colonies, no problems. What do you think? Cheyenne's got it. Brooke's got it. Sydney's got it. Good job. Obligate anaerobe. This sure looks like an organism that cannot survive when oxygen's around. This looks like an organism that oxygen is toxic to. Kills it. We got no growth when it was exposed to oxygen. We got nice growth when we took all the oxygen away. So this one looks very much like an obligate anaerobe. All right, we'll keep that in mind. Make a prediction for me. If we're right, and Clostridium is an obligate anaerobe, Where should it grow in the thioglycolate tube? What kind of pattern should we see in that thioglycolate tube? Where would these cells grow for us, if they grow at all? Where do you think you would see them? Brooks got it. Anybody else? Brianna's got it. Good. Way down at the bottom of the tube, right? In the anoxic zone and way at the bottom, where there's not a trace of oxygen, right? Remember, oxygen's toxic to this. If we're correct, oxygen's toxic to this organism. So it's going to tuck itself down at the very bottom of that tube. And then it should be able to grow fine because there should be no oxygen down there. Okay, good job. All right. So I'm going to keep this slide and I'll pull it up periodically so we can remind ourselves what should grow or what can grow with oxygen, without oxygen, catalase test results, and so on. I'll keep this slide available for us if we need it. All right. On the next couple of slides, we're going to look at the tubes. We're going to look at the actual thioglycolate tubes. Here's the Clostridium tube. So remember, this tube is full of thioglycolate and also a little bit of agar. It does not have any indicator dye in it, so we can't see where the oxic zone is, but we can estimate that it's at about the top 1/5 of the tube. Now, look down at the bottom. This is what I mean when I say cells clump together in these tubes. You see this little clump right here? It looks a white sort of cream color. Those are cells growing, and they've all clumped together into a little lump. This is the Clostridium organism, and it's growing just like we predicted. It can't grow up here. I know it's not growing up here because I don't see any cream-colored clumps or strings. I have to look down at the very bottom of the tube to see it. Okay, now that's not a lot of growth. You're going to see in the other tubes that some of these organisms grew into big clumps. So our Clostridium didn't grow super well for us in this tube, but again, it's very sensitive to oxygen. So we give it a little slack when we're using it in the lab, because if it gets exposed to oxygen, it's going to die. And sometimes when we're moving it around, some of the cells die from oxygen exposure. But we got some growth at the bottom of the tube, and that supports our hypothesis that this organism is an obligate anaerobe. All right, here's our second tube right here. Let me get the Clostridium out of the way for us. This is the Enterococcus Fecalis tube. So remember, this is a gram-positive intestinal organism. Take a look at this growth pattern. This is what I mean when I talk about a big clump of cells right here, sort of like a big mat, a big sort of collection of growth, all that cream-colored mass. And this is what I'm talking about when I talk about these strings. We don't see just nice cloudy media. What we see are these strings, these strands of growth. And it's the extent of the strand that you want to evaluate. So there are living cells in this strand, and they're growing and dividing and making this strand thicker and thicker. And as you can see, they're capable of surviving all the way down the tube, right? You see that? I've got cells growing down here where there's no oxygen. I have cells growing here where there's a lot less oxygen. And I've got cells growing up here where there's plenty of oxygen. But look at this. There are no cells growing way up at the top of that tube, are they? It's clear up there. So again, if I think about my oxic zone right here, this is an organism that can grow in oxygen, but it appears to grow in the lower portion of that oxic zone. Okay, so we've got Enterococcus Fecalis growing in the lower oxygen environment, lower amounts of oxygen, and the rest of the tube. Okay, so keep that one in mind. Jot that down for yourself. Let's look at the other ones too. Here's our friend E. coli. I actually have two tubes to show you for E. coli because E. coli did some interesting things. Let's look at this tube first. So here is growth in the oxic zone. Okay, and it's growth all the way through the oxic zone. Compare that to the last one we looked at, where we had growth in only the lower half of the oxic zone. Again, this is the growth, this big clump of white, sort of cream colored material. These are cells, and they're growing right here, but they're not growing up at the top of the oxic zone where the highest amounts of oxygen are. This organism is growing up to the top, all the way through the oxic zone. Now, it's hard to see, but can you see this white streak right here? This organism is also growing here. It's just not growing as nice as we would have liked right here. And that's not so much about the organism as just, you know, sometimes when you're doing experiments, they don't come out exactly like the textbook. Now, that's why I also wanted to show you this tube. You can see there's white material in the oxic zone. And I also have these strings hanging down. Okay, so this is an organism, this E. coli, that is growing throughout the oxic zone and down into the rest of the tube. Let me move these pictures around so we can look at the last one here. This is Alkalogenese. And if you look at the Alkalogenese Fecalis, this is where the growth is, right here, oxic zone. The whole oxic zone, but I don't see any other things growing down below. All right, so this one is growing only in the oxic zone, but it's growing all the way through the oxic zone. All right. So obligate anaerobe, like our Clostridium, limited to the bottom. Obligate aerobes grow only in the oxic zone, but they grow all the way through the oxic zone. Did we see any tube where we had an organism growing all the way through the oxic zone, but nowhere else? Here's these. Clostridium, Enterococcus, E. coli, Alkalogenese. Enterococcus, Clostridium, E. coli, Alkalogenese. Do we have an obligate aerobe, do you think? Yeah, we do. Which one? Type it in the chat for me. Where do we see an organism growing only in the oxic zone. No strings hanging down. Only in the oxic zone and all the way through the oxic zone. Brianna, let's take a look at E. coli again. This one sure looks like it might be just the oxic zone, right? But this is real. This string right here, that's real. There are cells hanging down from that area, growing down into the lower parts of the tube. So it looks like it's really happy in the oxic zone. It's growing really well in the oxic zone. That's where it's growing best. But it is also hanging down. And how do I know that for sure? Well, I have a second tube of E. coli. This one, you can't really see it as well growing all the way through the oxic zone, but you can see the strings in this tube much better. So this is an organism that can grow in places other than the oxic zone. So it's not E. coli. Yeah, Brooks got it. Brianna's got it. It's the Alkalogenese. It's the Alkalogenese. The organism that is growing only in the oxic zone is an organism that must require oxygen. It cannot grow below the parts of the tube where oxygen is present. It just can't because it needs oxygen in order to grow. It requires oxygen to make ATP. So it's an obligate aerobe. And that's the Alkalogenese. That's what we're seeing in that tube. Okay, good job. Now, what about facultative? Facultative anaerobes are the ones that are super adaptive. They grow best in the presence of oxygen. That's what all this heavy area of red indicates. But they can also grow in other conditions. They're happy to grow all the way through the tube. So do we have a tube where we see growth all the way through the oxic zone, heavy, and also strings hanging down? Here's Enterococcus, Clostridium, E. coli, and Alkalogenese. We'll leave Alkalogenese small right now since we already know what that one is. This one certainly is growing all the way through the tube, right? But it's only growing in the bottom of the oxic zone. This is our Enterococcus. So, and this one, of course, this one's only growing down at the bottom. So we can leave that one. But this one, you know, yeah, it is growing throughout the tube, but it's missing right here. And if you compare that to this tube, this one is growing all the way through the oxic zone. And it's got some also hanging down into the lower parts of the tube. It's our E. coli, right? That looks like a facultative anaerobe. It's going to grow best when oxygen's around. It's going to grow best when it's exposed to room air. But if there's no oxygen, like in the lower parts of that tube, it can grow there too. It just doesn't grow as well there. Good job. So we have two other categories. We have micro aerophile and we have aerotolerant left on our list anyway. And we have one other organism that we haven't classified, and that's this guy, the Enterococcus Fecalis. Take a look at how Enterococcus is growing. Do you think it's a micro aerophile? Or do you think it's aerotolerant? I've got nice heavy growth in the bottom of the oxic zone, so lower concentrations of oxygen. And I've also got growth down through the rest of the tube. What do you think? Pretend you're taking an exam, a lab practical exam, and I ask that question. What category, which one of those 5 categories do you think Enterococcus Fecalis belongs to? We already have one that's an obligate aerobe and one that's an obligate anaerobe, and we have one that's facultative, and I can tell you it's one of the last 2. Is it aerotolerant or is it a micro aerophile? What's the difference? What's the difference? Remember what the difference is between those 2. Aerotolerant versus micro aerophile. Translate the words if that helps. Brooks got it. Leanne's got it. Sydney's got it. This is an aerotolerant, right? Remember what the difference is between those 2 categories. A micro aerophile, think phile means love. Philic or phile means love. So it loves air, so it loves oxygen, but at micro levels. So it's not going to grow through the whole oxic zone. It's going to grow down in the bottom half of the oxic zone. And that's the only place it's going to grow, right? Because micro aerophiles require oxygen. They have to have it to make ATP. So you're not going to see any growth in the bottom of the tube for a micro aerophile. But aerotolerants are different. Aerotolerants tolerate air. They tolerate oxygen, but they don't need it to make ATP. So they're going to grow best in a low oxygen environment. So that's the bottom half of the oxic zone. But they're also going to grow throughout the rest of the tube. And Enterococcus turns out to be an aerotolerant organism. It'll grow all the way through the tube, but you're going to find it in the heaviest growth in the lower half of that tube. Good job. And that's just how we use these tubes. That's just how we use them. Let's say you get a sample in from a patient, and this patient has been suffering with some kind of an intestinal disorder, and the physician has collected a stool sample perhaps, or maybe taken a biopsy from the intestine, and this material has come to the lab, and they're trying to identify this microbe. One of the easy things we can do, right, is grow it, put it in some broth and grow it. But that's not going to identify what it is. We need some data about it. And one of the ways we can get that is by inoculating some of those cells down into that thioglycolate tube and putting that in the incubator and seeing how it grows. We're going to see what kind of pattern it grows in, and that's going to give us some important information. Most bacteria are facultative anaerobes. So if I get a result of facultative anaerobe, it's not really telling me a lot. I'm going to need some more results. Let's say the physician puts a note on the request for the lab and says that they suspect that this is E. coli. So I inoculate my thioglycolate tube and it shows me a facultative pattern. It shows me growth throughout the oxic zone, but I also see strings hanging down to the bottom of the tube. So I think facultative anaerobe, that fits for E. coli. But there are lots of other facultative anaerobes too. So what other media could I inoculate with that sample that would give me a lot more confidence that this was E. coli. What other media have we talked about, not necessarily in this lab, but in other labs? What other media have we talked about where we know we have E. coli because it grows in a particular way with a particular color pattern? What other media could I streak with that sample and put in the incubator that will give me a lot of assurance that, yes, this is E. coli? What do you think? That's right, Brooke. The green one, right? E. coli grows as purple colonies with a green metallic sheen. What was the name of that media? You can use the initials. What was the name of that one? Does anybody remember? That's a good guess, Brooke. Yep, EMB. EMB, it stands for eosin methylene blue. So everybody calls it EMB. That's the one that has that purpley sort of maroon color to the plate. And it'll grow a lot of organisms. It'll grow anything that's gram negative and lives in the intestine. But when E. coli grows on it, we get that really unusual green color. That tells us, oh yeah, that's E. coli. We can report that back. We can say, not only did it grow the way a facultative anaerobe grows, which we would expect for E. coli, but it also grew green, as shiny green metallic on EMB. Good job. All right, so the Clostridium Sporogenes, just to summarize, wouldn't grow for us in room air at all. Grew in the brewer's jar, though, and grew in the deepest anoxic regions of the thioglycolate tube. This is classic for obligate anaerobe. Alkalogenese, it grew happily in room air, no growth in the brewer's jar. It grew only in the oxic zone of the thioglycolate tube, obligate aerobe. Enterococcus Fecalis, well, it grew in room air and it grew in the brewer's jar, but it grew poorly in the brewer's jar. It grew small colonies in the brewer's jar. It grew in the lower oxic region of the thioglycolate tube and had strings hanging down into the rest of the tube. This is our aerotolerant organism. And finally, our E. coli grew in room air, grew in the brewer's jar, but again, grew poorly, and grew throughout the thioglycolate tube, but grew best throughout that oxic zone. This is a facultative anaerobe. And we didn't have a micro aerophile, but if we had one, we would have seen this. Growth only in the lower portion of the oxic zone. Micro aerophile loves air, loves oxygen, but only in micro amounts, low amounts. Good job. Questions? Questions about any of these categories or what it means for the organism? I sometimes have students ask about, sometimes a student will say to me that it doesn't really make sense that anything that lives in your gut could have a requirement for oxygen or could prefer oxygen because, you know, your gut is in the inside of your body. So it's not like it's getting exposed to room air. And you're right, it's not. It's not. But that doesn't mean it's an oxygen-free environment. This is a gross thing to say, but you have a tube that runs through you that's open to the air. It goes into your mouth, it goes down your esophagus, it goes into your stomach, it goes into your intestine, it goes into your colon, and it goes out your rectum. It's a tube, and it runs all the way through you. And a certain amount of oxygen is in that tube all the time. Okay, obviously you're going to have the most oxygen in your mouth because your mouth is open, really open to the air, but you will have oxygen throughout that tube. The only places in your body where you have very low levels of oxygen are really deep inside certain tissues. But of course, even in those areas, you're going to have some oxygen, right? Because oxygen is constantly being transported into cells. All of your cells have a requirement for oxygen because we are obligate aerobes. But there are places outside of your cells into the extracellular matrix, if you remember from anatomy, where oxygen can be very, very low, even almost absent. So yeah, it's a strange thing to think that you have oxygen down in your intestine, but you do have some. And that's why a lot of these organisms are very comfortable growing down there. All right, very good. All right, if there are no questions, I'm going to go ahead and switch gears now and get us at least started in our next exercise, which, if you remember, is about ultraviolet light. So you may or may not have watched the lecture yet about controlling microbial growth, but we generally use a couple of different things to control microbes around us and in our world. We use physical methods, we use chemical methods. Okay, and we're going to talk about both. We're actually we're also going to talk about the kinds of chemicals we use outside our body and the kinds of chemicals we use inside our body. So, chemical things like disinfectants versus antibiotics and other drugs that you would take internally to control microbial growth. In terms of the physical methods, the most common way that we control microbes around us is with heat. If you think about it, we cook our food, we use heat all the time to sterilize things, and even just to disinfect things. But there's another very common physical method that we use to control microbes, to stop them from growing in our environment, and that's ultraviolet light, or what's more properly referred to as ultraviolet radiation. Okay. I'm going to pull up the next set of slides for us. And again, we'll just get started on this today, and we'll continue with it on Friday. So ultraviolet is a type of what's called electromagnetic radiation, and it's often referred to just as ultraviolet radiation. We're going to look at the effects that UV radiation has on bacteria. And we know from previous work, that ultraviolet can do two things to bacterial cells. First of all, it can kill them. It'll kill them outright if they're exposed to it for a long enough period. And it can also damage their DNA without killing the cells. All right, we'll be talking about that in some detail. So one of our objectives is to learn about the mechanism. How is it that ultraviolet radiation penetrates into a bacterial cell and damages the DNA molecule? Because that's the molecule that it specifically targets in bacteria. We'll talk about how sometimes damaged DNA can lead to mutations in the cell. And we're also going to talk about a couple of repair mechanisms that are found in bacteria, yes, and also in other types of organisms. A couple of mechanisms that exist in these cells that can actually find damaged DNA and repair it. These mechanisms are referred to as photoreactivation, or what sometimes is referred to as light repair. That term photo, anytime you see that in a scientific term, it's referring to light. So light reactivation or light repair of DNA. And then there's a separate mechanism that sometimes is called dark repair, and that one is excision repair. Two different mechanisms, two different pathways that are inside bacterial cells that can locate damaged DNA and fix it. Let's take a minute now and talk about ultraviolet radiation. You know, we sometimes think that there's a difference between the light that surrounds us that we can see with our eyes and other forms of radiation. And the truth is, all of it is called electromagnetic radiation. And that's what you're looking at on this slide. So on this slide, you're looking at a spectrum from gamma rays, very, very powerful type of radiation that, you know, you associate this with nuclear power plants, for example, or nuclear bombs. Gamma rays over here on the left, all the way through a spectrum to AM radio waves, very, very weak type of electromagnetic radiation. And right in the middle, right about here, this little sliver that you can see, they've taken that sliver and they've made it big for us. This is where visible light falls in that spectrum. So what you and I can see, the colors of the light spectrum from purple all the way up through green and yellow and reds. This is a tiny slice of this spectrum of electromagnetic radiation. Now, remember from your physics classes that what makes electromagnetic radiation kind of special in the physical world is that it has this unusual property of traveling as both a wave and a particle. Remember when you learned about light and they talked about how light travels as a wave. It has the shape of a wave on an oscilloscope, but it also behaves like a particle, which is why we talk about photons of light. So those properties apply to this whole spectrum. So gamma rays have both wave and particle behaviors. And all the way through the spectrum, through X-rays and through ultraviolet rays and visible rays, all the way out into microwaves and radio waves. All of that is just types of electromagnetic radiation that have different wavelengths and therefore different amounts of energy associated with them. So remember, a wave, when you think about a wave, it has a peak and it has a trough, and then a peak and then a trough. And if we measure from peak to peak, or from trough to trough, from the top to the top, or from the bottom to the bottom, we're getting a wave length. And all the way through that spectrum, there are different wavelengths of electromagnetic radiation. The shorter the wavelength, the more powerful that radiation is. The longer the wavelength, the weaker that type of electromagnetic radiation is. So let's take a look at that spectrum again. Visible light has a wavelength of about 400 nanometers up to about 700 nanometers, a little bit past 700 nanometers, okay? So nanometers, from peak to peak, that wave of visible light, or those waves of visible light, range from about 400 to 700 or so nanometers, all right? And that's what you can see. Go down to the gamma rays. Now we're talking about 10 to the minus 14th. 10 to the minus 14th. Now this is in meters. So if we compare it to the visible light, we're around the area of 10 to the minus 5, 10 to the minus 6. versus 10 to the minus 14th. Very, very short wavelength for gamma rays, very, very powerful type of electromagnetic radiation, yeah? And of course, that makes sense because we know that this kind of radiation, this is ionizing radiation. In fact, gamma rays and X-rays fall into the kind of radiation that is ionizing. It can penetrate all the way through our bodies, all the way through living tissue. And as it penetrates, it's so powerful that it literally knocks electrons out of their orbit around atoms. Remember, electrons travel in those shells. And these waves are so powerful, when they bump an electron, they bump it out of orbit. So they create what we call radicals, free radicals, which are very damaging to living tissue. And of course, if you get exposed to too much of this kind of high energy, short wavelength radiation, you'll die from that. On the other end, if you look at the radio waves, FM here, here's television, here's shortwave, here's AM. Look at these. These are not 10 to the minus 6 or 10 to the minus 14. These are 10 to the 2 or 10 to the 4. 10 to the 4 meters long, these wavelengths are. These are very long waves. And therefore, they have very little energy associated with them. And as I'm sure you know, these kinds of electromagnetic radiation cannot penetrate through your body. They can't do it. They just don't have the power, the energy to do that. In fact, nothing on this side, all the way over to here, can penetrate at all. Radio waves can't penetrate, radar can't penetrate, infrared cannot penetrate, visible light cannot penetrate. But then we hit a change right here at ultraviolet. Ultraviolet has a wavelength of 10 to the minus 8th. So a shorter wavelength than visible light. If visible light's around 10 to the minus 6th, ultraviolet is 10 to the minus 8th. Shorter wavelength, more powerful. Not as powerful as X-rays and gamma rays, but more powerful than regular visible light. So it can penetrate. It can penetrate living tissue, but it's not ionizing. So when it penetrates, it doesn't knock electrons out of orbit. It doesn't cause free radicals to form in tissue. It's not powerful enough to do that. It also cannot penetrate all the way through the body. In fact, it has a really poor penetration depth. Ultraviolet can penetrate through living tissue to about 2 millimeters. 2 millimeters of penetration. And that's it. So think about what you know about ultraviolet radiation. It comes from the sun, right? And you know that if you go out in the sun for too long, you run the risk of getting a sunburn. You can get a bad sunburn, right? And even getting skin cancer. Your skin is susceptible to that. Your cornea are susceptible to that. You can get cataracts from ultraviolet exposure. But it's only your outer body that's susceptible because ultraviolet cannot penetrate into the inner parts of your body. So you can't get, ultraviolet exposure causing lung cancer or causing liver cancer or kidney cancer because it just can't get in. But your skin is susceptible. Now think about if you were a microbial cell. If you're a bacterial cell, bacterial cells we set in general, maybe 1 to 2 micrometers long. And there are 1,000 micrometers in 1 millimeter. Ultraviolet can penetrate to 2 millimeters. So ultraviolet can just go right through microbial cells. So they are much more vulnerable to ultraviolet than you and I are. You and I are vulnerable in terms of our skin and our cornea, but we are not nearly as vulnerable as those poor little bacteria are. And that's good because we can use ultraviolet to kill bacteria. Now remember, it's not ionizing. It's doing something different when it penetrates those bacterial cells. It's not knocking electrons out of orbit. It's damaging the DNA molecule. And it does something super specific to the DNA. Ultraviolet does. This slide just shows again, and this one's a little confusing because the gamma rays are over here now. They were on this side, but now the gamma rays are on this side. Here's the X-rays, ultraviolet, visible light, infrared, microwaves, radio waves. Okay, so we've just turned the spectrum around. Short wavelengths on this end where the gamma rays are, very, very strong, very energetic, powerful. Long wavelengths on this end, much less energetic, much less powerful. Anything over here to the left from visible light over cannot penetrate living tissue. Anything to the right from ultraviolet over can penetrate, but penetrates to different depths. All right. The last thing we'll talk about today is the different kinds of ultraviolet, because it turns out, just like visible light has a range of wavelengths, remember we said it ranges from the purple at about 400 nanometers, all the way up to the red at about 700 nanometers. Just like visible light has a range of wavelengths in it, so does ultraviolet. And ultraviolet ranges from about 15 to 400. Okay, 15 nanometers to 400 nanometers. And we can break this range of wavelengths into 4 different groups of ultraviolet. Most of the ultraviolet from the 15 nanometer up to the 200 nanometer range. Again, we're talking about wavelength here. That range, which is the most powerful, right? Shortest wavelengths, more energetic. That ultraviolet radiation doesn't even reach the ground. Most of it gets absorbed by molecules that are just floating around in our air. So lucky for us, most of that doesn't even reach the ground. But between 200 and 400 nanometers will come all the way down to the surface of the Earth. So let's start at that 200 end, 200 to 290 nanometer wavelength. This is called UVC, UVC. And this type of ultraviolet is what we call biocidal, cidal meaning death. So death to living things when they get hit in this range, because at this range, the ultraviolet radiation is optimally absorbed by the DNA, and you get the most damage to living cells. So ultraviolet UVC rays are the kind that can kill cells. Now I have a reference here to a manual. Just ignore this because we're not using our manual this semester. So that's 200 to 290. 290 to 320, so a little bit longer wavelength. This is what we call UVB. UVB can also damage DNA, but it's not in the biocidal range. Something to note too about UVB. UVB exposure to a degree, is actually good for us, because right around 300 nanometers, that wavelength is the wavelength that we need for our skin to convert vitamin D1 to vitamin D3. That's the active form of vitamin D3. So you've probably heard that, being out in the sunshine is actually good for you a little bit, because it helps your body make vitamin D. It makes the active form of vitamin D. Now, the last group is UVA. That's in the wavelength of 320 to 400. UVA rays are not readily absorbed by DNA. It's just not the right wavelength. Now, UVA can still damage your skin and your eyes, but not through DNA damage. So UVA rays can create, the wrinkles that we hear of from too much skin exposure. They can certainly contribute to cataracts. In other words, they can still damage your skin and eyes, but it's not through DNA damage. Now, the next time you go to the store and you buy your sunscreen, look at the bottle, because the bottle by law has to tell you which rays it's capable of absorbing, basically blocking. And when you read about a sunscreen that is full spectrum, they're talking about a sunscreen that can block UVA, UVB, and UVC. A lot of people don't like full spectrum sunscreen, because guess what else will happen when you wear a full spectrum sunscreen, at least if you use it correctly? You won't tan. So a lot of people don't like full spectrum sunscreens, but they are the best protection for you against, again, DNA damage in your skin, which can lead to mutations and cancers. All right, very good. We're going to pick up here when we get together on Friday, and we will talk through another experiment. We will expose some bacteria to ultraviolet radiation, and we'll see what happens. We'll see what happens to them when they get exposed, if they can survive it. And if they can survive, can they repair the damage that's been done? Okay, and that'll be on Friday. All right, everybody. Good to see everybody. Enjoy the rest of the afternoon, and I will see you on Friday.